So our next and final speaker for this morning session is Dr. Sharon Plon from Baylor College of Medicine. She's an excellent speaker with an extensive CV. I'll be highlighting only some key facts from that CV. She's a diplomat of the American Board of Medical Genetics and Genomics. She received her MD and PhD in biophysics from Harvard University and completed her medical genetics training at the University of Washington. She is a professor in the departments of pediatrics, hematology and oncology, molecular and human genetics, and human genome sequencing center at Baylor College of Medicine and Texas Children's Hospital. She serves on the National Advisory Council for Human Genome Research of the NIH, and she serves on the board of directors of the American Society of Human Genetics. She led the NHGRI NCI funded basic clinical trial of whole exome sequencing for newly diagnosed childhood cancer patients, and this study is now being expanded into several different centers at Texas. She also co-chairs the Germline Reporting Committee of the Children's Oncology Group and the NCI Pediatric Match National Precision Oncology Trial, which opened in August 2017. She is also a principal investigator of the NIH-funded Clinical Genomics Resource and chairs the ClinGen Hereditary Cancer Working Group. She has extensive experience in the education of students and physicians, and since 2006, she has directed the Medical Scientist Training Program at BCM. Without further ado, Dr. Plon.
Well, really nice to be here, and I've actually never been at Mardi Gras before, so that was a big surprise when I arrived last night. So I knew that the first two speakers were going to talk a lot about germline analysis in a variety of different settings. So I actually am going to particularly focus on the setting of childhood cancer patients. I should say that almost everything I'm going to say is true of adult cancer patients as well, but I'm really based in a children's cancer center. So what is really the problem facing pediatric oncology? In some ways, we suffer from our great success. People think the problem's been solved. 80% of children with acute leukemia can be cured, but there are still many pediatric tumors with really very poor outcomes. And I'm going to use pediatric brain tumors as kind of my example throughout the talk. And this is just an example of an unfortunate child, a 12-year-old boy who had a glioblastoma, treated standardly with surgery, radiation therapy, and chemotherapy. But again, despite, and you can see the cavity from the prior treatment, but where that red arrow is pointing is unfortunately what we almost always see in the case of GBMs, which is that the tumor had recurred. And at that time, there were really no good treatment options, although I'll come back to this patient. But then, as I'm a medical geneticist, it's also true that we don't really have a good sense of what proportion of childhood cancer patients actually have an underlying genetic susceptibility to cancer. And that might not help them with regard to their treatment, although I'll give an example where it does, but it tells us a lot about the risk to family members. So that's kind of the problem. So I want to just briefly, very briefly, go through the history of what I would call the beginning of cancer genomics. This is a paper by my colleague Will Parsons when he was a fellow at Bert Vogelstein's, and it's actually the first paper where cancer genomes, in fact it was exomes, were sequenced. And for those of you who are laboratory scientists, this was actually Sanger sequencing. So this was right before next generation sequencing became possible. So they only did 10 tumors. But from those 10 tumors, they actually identified a very unexpected mutation in two related genes as part of the Krebs cycle. IDH1 and IDH2, and these missense mutations were almost identical in these two tumors. They went on to sequence some other tumors for this gene and found that it was recurrent. And in fact, we now know that IDH1 and 2 mutations occur in AML, a variety of sarcomas, but again, no one would have looked at this gene without doing whole exome sequencing. Fast forward 6 years, just to show you how rapidly things have changed, and this is now a large paper published in 2014 that shows next generation sequencing. I think this paper included both DNA and RNA sequencing of now a wide variety of patients with pediatric brain tumors, high-grade gliomas. And you can see how many different genomic findings we now are able to categorize in each of these. And it just gives you an idea how rapidly we've been able to characterize these tumors. But all of what I've showed you up until now was research sequencing, not sequencing that was, these are compendiums of accumulated tumors from patients, not sequencing that we were doing anything about. So I didn't realize that Matt was going to give that incredibly elegant definition. So I'm going to give a less elegant definition of precision oncology, which many people define as the tailoring of cancer prevention and treatment based on the characteristics of each patient. It, of course, is not a new concept. We've done cancer cytogenetics and a wide variety of other methods to characterize tumors. Generally, when people talk about prevention, they're talking about germline analysis, the analysis of the blood, and that's what I am mainly focused on. And treatment, of course, generally requires some direct analysis of the tumor. So before I launch into some of our research, I do just want to say, and to kind of go from 2014 to 2018, is we do already have some success stories. So the most recent success story is this paper that came out in 2018 in the New England Journal. Larotrectinib was actually a drug specifically developed to target fusions. So these are fusions that occur at the DNA level, but you make a fusion protein involving the TRK kinase. And these fusions are actually found at an extremely low level in a wide variety of patients. But a startup biotech company, which my understanding was just bought for several $billion, went out to find these patients, and they developed a clinical trial design where they would test any tumor, and they were eligible. The study was eligible to essentially anyone in the country who had a TRK fusion positive tumor. And so you can see the wide variety of tumor types, because overall it turns out only 1% of tumors contain this fusion. But if you are one of those fortunate patients, this drug was incredibly effective in decreasing the growth of those tumors. And in fact, I believe in October, just a few months ago, the FDA approved this drug specifically for any tumor with a TRK fusion. So that really gives you kind of the idea of where we've come from 2018 with regard to research. But if we're really going to do precision oncology, then we really need to be able to do clinical grade genomic testing, and we have to be able to implement it. We have to do research on how do we implement it in a real clinical setting. So with that introduction, I'll just get to a couple of the main projects we've been involved in, and Bruce actually is now involved in the second phase of this. What was originally called the Clinical Sequencing Exploratory Research Consortium, or CSER, was designed by NIH pretty unusually to say, we just want you to do genome or exome level testing in a clinical setting, in any clinical setting, and tell us how it works. That's essentially what the RFA said. You didn't have to prove anything. You had to prove you could do it, and you could measure what happens. And so 9 sites were funded, 3 of which actually proposed to do both tumor and germline sequencing. Ours at Baylor College of Medicine that I'll talk about in children. University of Michigan that did both children and adults with very high-risk tumors, including a lot of sarcoma patients. And the Dana-Farber that did adults with metastatic, I think, colon and perhaps lung cancer. And then the University of Washington actually did adults at risk for colon cancer. So we had kind of a number of groups that wanted to specifically look at, can we do genome or exome level sequencing in a cancer setting? This is our team at Baylor. I do want to just highlight again Will Parsons, who's now been my co-PI for 6 or 7 years. He's an outstanding pediatric oncologist who really has led the tumor aspects of the study where I've done the germline. I also just want to say it's really been one of the great experiences in my career. One of the nice things about being in academics is you get to do different things. To do this, the Genome Center run by Richard Gibbs and Christine Eng, a very experienced molecular diagnostician, worked together with a whole large team of us to develop a lab that would do clinical exome sequencing, both for cancer patients and actually primarily for non-cancer patients, and make that available to the community. We also worked with Amy McGuire in the Center for Medical Ethics throughout the study. So our study was called BASIC, it was actually CUBE, that's our old slide, but everyone calls it BASIC 3, for Baylor College of Medicine Advancing Sequencing into Childhood Cancer Care. And the bottom line is we open the enrollment to any newly diagnosed solid tumor patient. So CNS or non-CNS solid tumors. We tried to get a frozen specimen at the time of diagnosis and a blood sample. And if the families consented, then the blood and tumor were sent to the clinical lab I just described, where CLIA-certified whole exome sequencing was performed. A report that goes back into the medical record, just like any other test, was returned to Texas Children's Hospital, a separate report for the blood and the tumor. These went into the electronic medical record. They were returned to the pediatric oncologist, and then a genetic counselor and the pediatric oncologist returned the results to the family. And we followed these families for 2 years with a variety of surveys and interviews to really understand the impact of this exome on the families and the physicians. I should say in all of our studies, the oncologists are also subjects and do a wide variety of surveys and interviews. The methods have all been published now a couple of years ago, but I'm going to give the updated, when we were about halfway through the study, I'm going to give more of the updated results. So the first thing, which was brought up earlier, is the importance of diversity. We're very fortunate. Houston is considered one of the most diverse cities in the United States, and it was very important to us that the families in the study represented our patient population. We were very gratified when we looked at the first about 120 families that were approach to enroll, that there was no difference. Again, this is now the data, this is modified from the paper. This is now the data for the entire study, and there was absolutely no statistically significant difference in who decided to enroll or decline based on whether they were Hispanic or non-Hispanic, and there was no statistical difference with regard to race. And so we were gratified that we were able to discuss the study with our families and get fairly uniform enrollment. Overall, 70% of families that were approached agreed to participate. And when you ask why did they decline, again, this is earlier data when it was at 80%, but these numbers haven't really changed. It was not what we were first concerned about. The main reason people declined was not concern about genetic risk or privacy. The main concern was they were just overwhelmed. Their kids were just diagnosed with cancer. They might be on a treatment trial. There was a ton going on. They didn't necessarily want to be involved. And even that was still just a minority. But again, until you ask families, you might get different responses than you as a physician expect to begin with. The other aspect of diversity that makes pediatric cancer challenging is the diversity of the tumor types. Unlike adult cancer, where there's several very common malignancies like breast cancer or colon cancer, pediatric cancer fortunately is rare, and there are a very wide variety of tumor types, which pretty much represent, this pie chart represent what you would expect for the pediatric cancer patients in the general population. So then, of course, if you're doing exome or genome testing, what people often don't talk about is you have to decide what are you going to report. There are actually all kinds of things you can report. And in this study on the tumor side, and people also filter differently, what we decided on the tumor side was to essentially report everything. So every somatic mutation we found in a gene, in a tumor, was reported back to the doctors. As you'll see, we tiered them with how important they were, but we knew that the knowledge was changing incredibly rapidly, and we didn't want to filter something out that 3 years from now when the child relapsed turned out to be something that was important. And so I'll talk about that first. So this is a classic diagram from the Vogelstein lab when they first did sequencing of tumors, and these big mountains were what were found in adult cancers, where you have certain genes like P53 or MYC that are incredibly common. Turns out you have very few mountains in pediatric cancer. But we did find that some genes like beta-catenin and P53 had mutations, and even BRAF had mutations in about 5 to 10% of patients. There were another 2 to 5% that had these genes. So these genes are recurrent, but across 300 kids, that might still mean you only see them 3 or 4 times. And then there's a very large tail of mutations only seen once across the entire cohort. So this is important, again, when you think of the TRK inhibitor drug. We're going to have lots of tumors. We're going to have lots of genes we might be able to target, but may be found in only 1% or less of children. And that, with cancer, and that really mediates the MATCH trial I hope to talk about at the very end. Overall, there were clinically significant tumor variants in about 30% of tumors. And to go back to this original example, this patient was not in our study, but was published around the same time. This child with a glioblastoma actually was one of those kids with a BRAF mutation that we found in about 2 to 5% of our patients. There is actually a drug, FDA approved for that in adults. And in this study, they actually used that drug in this patient and actually had at least a good early response. It, of course, can be much more complicated. This is a child from our study with the complete tumor exome results, where this child with medulloblastoma actually had 5 different known cancer drivers that were identified in the tumor, which then makes decisions about treatment much more complicated. Of course, what I'm interested in is their germline findings. What do we actually find in the blood? And again, we actually reported all of these categories of results, other medically actionable findings, a small number of pharmacogenomic findings, and recessive carrier status that I won't talk about today. I'm going to just focus on how often did we discover. This was the fundamental question I was interested in. If you take unselected childhood cancer patients, how often do they actually have a cancer susceptibility mutation? And the answer turned out to be about 10%. And I should say that St. Jude Children's Hospital published a beautiful retrospective study, theirs was not prospective, where they also came up with about 8%. The Michigan Caesar Project published a paper around the same time as us that came up with 9% or 10%. In adults, there have been several papers that have come out between 13 and 15%. So this seems to be a pretty constant number, that if you just take unselected cancer patients and thoroughly look at cancer susceptibility genes, anywhere from 8 to 15% of them actually have a finding. And what struck us, so this was 27 of our 278 patients who completed the whole trial. Remember, we were aiming for 280. Of these 26 patients with a dominant cancer susceptibility mutation, it was 19 different genes. So again, just like the tumor, there were no big players here. This is different than the St. Jude result where they had a very selected patient population. P53 was only seen 3 times, as was VHL. And the other thing that was very striking is some of them were genes we knew caused a risk of that tumor, even though we might not have predicted it. So for example, both children with the Wilms tumor 1 gene had Wilms tumor. Neither of them had a family history of Wilms tumor. Neither of them had bilateral disease. And in both cases, it was de novo, or in one case, de novo mosaic. But there were another whole group of patients who had clearly dominant pathogenic mutations in genes that had not been associated with that child's cancer. We had 3 patients with BRCA1 and 2 mutations, PALB2, again, another adult cancer gene, CHEK2, and then we had some pediatric genes like SMARCA4, but in the wrong tumor type. The other thing that really disappointed me, because I've spent much of my life studying rare recessive cancer syndromes, is that they're really rare, even in childhood cancer patients. So we only found 1 recessive diagnosis in a child with liver tumor. Similarly, the St. Jude study, which was much bigger, I think of 1,100 patients, they found 2. So it's really true that these rare recessive disorders have a very small impact on the overall cancer risk. The final thing that I don't have time to talk about is we prospectively predicted for each patient at entry, would we do genetic testing? And if so, what gene would we test for? And we were extremely broad in saying who we would do testing for, and even so, only 45% of these 28 patients would we have actually recommended testing for the gene that we found. So our current guidelines for who should have testing really are insufficient. Why does this matter? This is a recent paper from David Malkin's group describing the Toronto protocol. These are children who were found to have P53 germline mutations, where we now do a very extensive screening for cancer. It's called the Toronto protocol after that group. And this was an update of their data showing that mortality from cancer, this is mortality, overall survival, from cancer was substantially better in children and adults who had had dedicated screening. So finding these patients, and at least finding their siblings and doing screening, really is now being shown to reduce mortality. And so this really has led the field to a challenge, because more and more children are being diagnosed with these syndromes. There are more and more guidelines out there, but there's no place to find them. So I have to really give the American Association of Cancer Research huge kudos. They had an international workshop that I helped organize back in 2016 They created a completely open access site. If you just Google pediatric oncology series, this will come up. And there are now something like 14 articles from experts in the field that describe the recommendations for, I think, something like 50 or 60 different cancer susceptibility syndromes. So if you have a patient in your clinic diagnosed with one of these, you have a handy reference to what to do about it. I always like to give an example, and to go back to pediatric brain tumors in GBM. Very early in our study, and that's relevant, we had a 14-year-old girl with a glioblastoma. The mother was aware of a substantial family history of cancer, although that was not reflected in the electronic medical record, and the child was found to have a Lynch syndrome mutation, an MSH2 mutation. That's actually known to be associated with glioma risk, although not typically in teenagers. And certainly this finding had big effect on the family. There are multiple members of the family diagnosed with Lynch syndrome who are now having screening. The child unfortunately went on to die of her disease. But if instead of being one of the first patients in the study, if she had been one of the last, it might have actually impacted her treatment. We now know that patients with Lynch syndrome respond extremely well to checkpoint inhibitors. In fact, the FDA has approved some of the immune checkpoint drugs specifically for mismatch repair defective tumors, and there are now actually clinical trials for children with brain tumors who have these kinds of mutations. So it does show how our knowledge evolves over time. I don't have time to talk about it other than to say that we've published. We did a lot of interviewing of the parents. What did this mean for them? Their sense of the utility of the data is very different than what an insurance company might think about it. There was a lot of interest around the issues of guilt, and we have a paper that I think now is in press that Janek Malik led, really looking at issues around parents and their feeling about guilt when their children are diagnosed, and how getting a genetic diagnosis or not getting a genetic diagnosis might impact that. They had a lot of feelings about using this data really pragmatically for future planning. Just briefly, we are now in our second phase of funding. We've launched the Texas Kids Can Seek study that has this incredibly long name. We now are branching out to now 6 different sites in Texas. The emphasis in the second phase of CSER funding is on diverse patient populations, so we're aiming for 60% diversity across our study. It's also diverse in terms of we're now looking at academic programs, non-academic programs, big programs, and small clinics. And we're also now directly comparing more focused hereditary and tumor panels with exomes and transcriptomes. And we really want to now learn something about which of these tests are most useful for the care. We're now really going to score how often do you find a mutation where the doctor actually makes a treatment decision. We're also really working hard, similar to what Bruce just talked about, to make this understandable and acceptable to families. We have a whole series of videos in both English and Spanish that we provide the parents. But what I really want to end on is the pediatric match study, because I think this is a good example of how far we have come. So this is a study, a precision oncology trial that is funded by the Children's Oncology Group and the National Cancer Institute. The objective is to open it across the Children's Oncology Group consortium-wide as a single-stage phase 2 trial of genomically directed therapies for children with refractory, so refractory means they've failed therapy, they've relapsed, or they've recurred tumors and lymphomas. So the patients do have to have a biopsy. So we've actually come that far. That was a big issue early on. Would parents agree to a biopsy at relapse for a research study in a child? So we do now feel there's enough data to warrant that. The child has to have a biopsy at relapse. That tumor is then undergoes genetic sequencing. We look for a specific list of actionable mutations. We then have an entire series of study agents that I'll describe where the patient may then be put into a treatment arm, and then we're assessing the response. So this has to be practical because we're doing this across the country. So we're using FFPE, this is no longer frozen tumors. It's at relapse. It's being processed in one site, so it goes to the COG Nationwide Children's Hospital site. And then there are 3 different match laboratories that are running exactly the same platform, and they're using a shared analysis platform. Again, RNA fusions are very important for treatment targets, so we're doing both DNA and RNA mutation and fusion panels. What I'm particularly proud of is that we spent 2 years convincing NCI and COG to do blood at the same time. The adult match trial, which is now almost finished, did not do any analysis of hereditary nature. You just got the report back on the tumor. Those are reported by 2 laboratories that are familiar with hereditary cancer testing, and we hope that it's going to provide information on genetic susceptibility to cancer. So this is really the power of this kind of match trial. This is as of like February. These are all of the arms that are open. So these are all different drugs. The TRK inhibitor actually was opened. I think it actually may close again because it's now FDA approved, but it's not FDA approved in children. So we try to not do drugs for which there's about ready to be a big specific study for them. But you can see all the different, I know you can't read it, but there's TRK, there's MEK, there's ALK, there's BRAF. These are all different inhibitors, some of which are known to work in adults but have not been tested in children. And then you can propose new drugs. So I was particularly struck that there is now an IDH1 inhibitor. So Will Parsons is the national study chair of this study. So 11 years after his original paper, we're now going to try to test an inhibitor of that mutation in children. So just to wrap up, 10 years after the first tumor exome, we really do have the early realization of precision oncology for childhood cancer. We do now have a nationwide accessible genomic marker indicated treatment trial for children with really the worst prognosis. And the tragedy of this is that, of course, these drugs don't all work, and not every child, in fact, the majority of children don't have a match. So we're really just at the beginning. But we've now already enrolled almost 467 patients. It's open in more than, I think it's almost 100 sites now around the country. And we're now enrolling about 30 patients per month. As I mentioned, as new drugs come online, new treatment arms can be updated, and actually patients can rematch. So we have weekly phone calls where we all get online. There's an encrypted web-based system where we can look at the results. I'm particularly involved in the germline results. But if a patient doesn't have a match or they fail a treatment arm, they can actually then be rematched. And it's really the first national trial to do matched tumor and blood analysis so that the germline status is also returned to the oncologists and their parents. And we hope that this is providing some useful information for families, even if a treatment arm is not available to them. It might identify relatives at increased risk that could undergo screening and prevention. And with that, I just want to thank the big team at Baylor that was involved in the BASIC 3 study. Thanks.